Hello students, welcome. This is module 2.2.2, Passive Processes, Simple Diffusion. The way that things move across cell membranes, we can categorize them into two main categories. First, passive processes. Second, active processes. And we'll describe what each of these means for you in turn at the appropriate time. First, we're going to discuss passive processes. These are processes that rely on pressure or concentration differences in the process of diffusion. Primarily, we're going to be talking about concentration differences or concentration gradients and how substances will then diffuse down such gradients. Now, the thing about those gradients is that they have energy, and therefore this type of transport requires no input of energy. It's not that it doesn't require any energy, it's that all the energy that is required is already there in the concentration gradient. And so this is kind of like a river in that respect. Moving downstream, you don't have to supply any energy, and transport occurs as you move downstream. The energy is there in the stream, just as the energy is in the concentration gradient. We are going to study three different types of passive processes: simple diffusion, osmosis, and facilitated diffusion. This module is about simple diffusion. Before we go on and discuss simple diffusion, we want to learn about some terminology, some concepts that are important that underlie that idea. And so first we're going to learn about solution, solvents, and solutes. A solution, we can write it as an equation, consists of both solvent and solutes. The solvent is the liquid portion, and solutes are what is dissolved in the solvent to produce the solution. Often in biological systems, the solvent is water, and the solutes are various water-soluble chemicals. And so there. The substances that are dissolved in the water then are the solutes, as I said before. And most transport inside cells occurs in solution, so to have some idea of this concept is of critical importance. We can also define for you the term concentration. Concentration may be defined as the density of solutes, or alternatively as the number of particles of solute per unit volume. And concentration is typically used to describe a solution. For example, if we had the same volume of water, 100 milliliters, and we put 5 grams of sugar in one such solution, and then we had another 100-gram beaker of solvent, and we put 10 grams of sugar in there, the second one would be twice as concentrated as the first, just as an example. Here's a pictorial depiction of a concentration gradient, and this is what we often see in cells, maybe not so grand as this. Over here we have C1, the area of greatest concentration, and C2, the area of lowest concentration, and intergradations in between. Often in cells we might have a cell membrane where there is one concentration inside the cell and a different concentration outside, but it still functions as a gradient. And remember, there is energy in the gradient, and that is how passive transport works. It takes advantage of the energy inherent in the gradient. So now that we know a little bit about solutions, solvents, solutes, and concentrations, we're ready to discuss the idea of simple diffusion. The definition of simple diffusion is the movement of a substance from an area of greater concentration to an area of lesser concentration. As you can see in this simple example here, we can see that the dye pellet is the area of greatest concentration with varying concentrations around it. And over time, because of simple diffusion, the concentrations will even out, and we will have the dye evenly dispersed. And so that's what occurs in simple diffusion. Typically, in simple diffusion, we don't talk about a membrane. There is no membrane in the example with the dye pellet. Here is really a depiction in the lower left corner, an animation showing the purple particles, and as they spread out, and then it stops. So we can see also the illustration here panels A, B, C, D. We put in a lump, in this case, of sugar, but it could be a dye pellet as well. And then the individual molecules or atoms disperse evenly because of simple diffusion, and they end up in what we call a dynamic equilibrium. Here is yet one more depiction of the same idea. In this case, we're putting a drop of dye in, and again, that's a very concentrated state of molecules, and they slowly spread out. And you can see the water there. We're actually showing the water molecules in this one. There are several points to remember about diffusion in general. Number one is that in diffusion, the molecules show net movement down the gradient, that is, from an area of higher concentration to lower concentration. And so that's the first point to remember. The second point is that the sharper or the greater the gradient, the faster the diffusion. A third point is that diffusion is only effective over a short distance in a short time. And the last point about diffusion is the end result is what is called dynamic equilibrium. And what this means, it means that at the end state, that molecules don't stop moving. It just means that their movement offsets one another. For everyone that moves to the right, one moves to the left. For every molecule that moves up, one moves down. And so that the net concentrations don't change, although movement continues. This is showing diffusion across a membrane, and there doesn't have to be a membrane present for diffusion to occur, but this is showing a membrane. And so we can see, in this case, molecules start at the left, and they end up, and over on the right, they finally equal out. And when they do, we have dynamic equilibrium with a similar number of particles. Now, again, remember that those particles or molecules don't stop moving. It's just for every one that goes from left to right, one goes from right to left, and so that the net concentration doesn't change. And they show below another series of panels with two different substances diffusing. What causes diffusion? This is an important idea to consider, and it was actually discovered, at least in part, by a botanist named Brown. And Dr. Brown was putting some pollen under a microscope to examine, and he observed the driving force of diffusion. The driving force of diffusion is random molecular collisions. And the way that Dr. Brown, the botanist, saw these collisions, he couldn't see the water molecules, but he could see the pollen grains, which are big, and they were being buffeted about by the water molecules. And he could not figure out what was making these pollen grains dance back and forth. And eventually it became known that he had discovered this driving force of diffusion. It is in fact named after him. It's called Brownian motion, the inherent motion of molecules that leads to collisions. And he couldn't see the water molecules with his microscope, but he could see the larger pollen grains that were being moved. And so this is using a super microscope showing water molecules showing Brownian motion. In fact, if we see this in the lab, we often put dye in there that has large particles, and these bouncing water molecules will cause the dye particles to vibrate, and we can barely see the dye particles with our microscope. So let's kind of let you know the size that we're dealing with there. In any case, this concludes our second module about cellular transport, and we will continue this later.